Hello and welcome back to SPARCS 18. It is my pleasure this afternoon to introduce you to Dr. Elinor Karlsson. Elinor is an assistant professor in bioinformatics and integrative biology at the University of Massachusetts Medical School. She is also the director of the Vertebrate Genomics Group at the Broad Institute of MIT and Harvard. She's excited by the potential for using our own evolutionary history to understand how the genome works and in how that knowledge can lead to advances in healthcare. Elinor has a special interest in diseases shared between humans and dogs. She recently launched the citizen science-driven Darwin's Dogs Project, which invites all dog owners to participate directly in research exploring the genetic basis of dog behaviour, as well as diseases such as OCD and cancer. Her current projects also include 200 Mammals Genome Project, an international effort led by the Vertebrate Genomics Group at the Broad to compare hundreds of different mammalian genomes and identify critically important segments of DNA. Please join me in warmly welcoming Elinor to SPARCS 18.
Thank you very much for that wonderful introduction. I'm really excited to be here. So as was mentioned, I actually work at two places that are really focused on human health. So it's kind of interesting that I'm here talking about dogs, and I'm not actually connected to any of the veterinary schools. And part of the reason for that is that I study genomes. And this is something, the DNA that we inherit, we actually share a lot of it with other species. And so by studying other species, we can actually learn new things about humans, but also learn about things that humans don't do. And one of the most fascinating species to study actually are dogs, because there are so many of them living with us. I had a graduate student a few weeks ago ask me, why do I study dogs? And I said, well, that's actually really easy. It's because there's like millions and millions of them in this country alone. I think there's something like 70 million dogs within the United States. And that's just the United States. And there's no other species where we have that many individuals. And each of them is living with a person, and that person knows so much about them. And they take their dog in for medical care. We've got veterinary records. And so it's really kind of, if you're trying to understand how genetics influences health, dogs are kind of a place to start that is going to offer you more opportunities than almost anything else. So I put this in. This is a picture of my lab. We do a holiday card every year that we have a lot of fun with. And since we don't just study dogs, we study many other species, including this 200 Mammals project we're doing. We decided to have a holiday card where we actually showed ourselves doing saliva swabs on imaginary species. And so we've got Cerberus, the three-headed dog, in there. I think we've got we've got a chupacabra in there somewhere, although I think it's technically a beagle cabra. And so we actually had a lot of fun with that. And it was just kind of to illustrate all the kind of crazy stuff we're doing. I just want to put a caveat in at the beginning of this. I study dogs. I've never actually owned a dog. I actually got into dog genetics because the person I was working with at the time, Kerstin Lindblad-Toh, came to me and she said, how would you like to work with this kind of dogs and coat colors for your PhD? And I said, that sounds so cool. So I got into dog genetics because they're a fantastic genetic model. And ever since then, I've been kind of learning all about them as a pet. And it's actually been a lot of fun. And since then, my sister's gotten dogs, and she's actually now a dog trainer, and she works in animal behavior, and it's been a lot of fun. But I am still a cat person. So I just have to admit that. So yes, I have cats at home. That's my cat, Lacey. She does get herself into a lot of trouble. But professionally, we study this species that we've been talking about all day, Canis lupus, and the subspecies Canis lupus familiaris, or pet dogs. And the reason that we're kind of so interested in studying them is partly because they have this really unique population history. So if you actually look into it, wolves and dogs, as I said, they're the same species. Dogs are a subspecies of wolf. One of the ways that we define species is whether two groups of animals can interbreed, and dogs and wolves have no problem doing that. There's a lot of people studying dog domestication. It's not something that I've focused a whole lot on, but I think I kind of went through the literature, and I think right now we've come to a consensus that dogs were probably domesticated somewhere between, maybe 12,000 and 40,000 years ago, that it happened in Europe, or the Middle East, or Central Asia, or East Asia, or maybe Africa, and that it either happened once or more than once. So totally got that covered now. What I've been spending a lot of my career studying since I talked to Kerstin about dog breeds all those years ago is actually purebred dogs. And purebred dogs as we think of them today, are actually not very old. And so the vast majority of breeds were created within the last 100 to 200 years. And this is kind of the time point in which we started, we defined a breed as a group of dogs, and only dogs within that breed got to be in that breed. There were probably things around before that, they just weren't actually kind of these closed breeding populations. And geneticists have been interested in studying breeds for a very long time because they have kind of these unique features, including strong selection, especially on physical traits like size and coat color and morphology and things like that. There's less genetic diversity within a breed that made it kind of easier to try and figure out what was going on. And in some of the breeds, we see very high rates of particular diseases. And so if we want to study diseases like, say, bone cancer, it makes sense to focus on breeds where they have a very high rate of bone cancer because it makes it a little bit easier to find the genes that are responsible. Note I said easier and not easy. Cancer is still really hard. But what I'm actually really interested in right now is something we haven't studied a whole lot, which is the 15,000 plus years between domestication and the breed dogs that we have today. And so the last couple hundred years we have the breeds, but before that it wasn't like nothing was happening. This was the time period when dogs were adapting to living with humans. They probably started eating our garbage. They adapted to our food sources. And at some point in time, humans probably did start breeding for dogs that had particular behaviors or traits that were useful to them. And all of that's happening long before the kind of recent breeds were created. And today we have these populations of dogs where we have these kind of behaviors that have been captured within the breeds. But behavior is complicated. It's controlled by a lot of different genes. And because of this complexity, we think that probably the actual selection on these traits was happening thousands of years before this. And we also have, as I said, kind of way back at the beginning of all this, I study at places that are focused on human health. And so a lot of what we're interested in is understanding the genetics behind human diseases. And one of the things that is kind of different about dogs, is actually probably not different about dogs, but dogs do suffer a lot of the same diseases that people suffer from. So things like, in the kind of behavior world, things like aggression, anxiety, cognitive dysfunction, age-related cognitive dysfunction, phobias, impulsivity. These are all things that have kind of direct matches in human medicine as well. And so we're hoping by understanding them in dogs, we'll actually learn something about the diseases in humans as well and be able to come up with new therapies that are going to help both species. And of course, it's not just the behavioral disorders, it's things like allergies, cancer, and many other diseases like epilepsy and autoimmune diseases as well. And so to kind of summarize the focus of my work and a little bit of what I'm going to talk about today is that what we're trying to understand is how do changes in DNA, which is just this kind of string of A's, C's, G's, and T's, actually lead to changes in dogs' behavior and health. I mean, I kind of remember when I kind of first started learning more about dogs and retrieving, people have these dogs that are, retrieving breeds. And then, these dogs will, many of them retrieve balls for hours on end. They're completely obsessed with it. And so my first question was, is how is retrieving a ball for hours on end not an obsessive compulsive disorder? That's a different question. But at the same time, how do you actually change DNA to create that behavior? And it turns out it's complicated. I think Claire mentioned this morning that even though we think of golden retrievers, for example, as being retrieving dogs, there are many golden retrievers that aren't interested in retrieving. It's a complex trait, and there's variation within these populations as well. And then by finding the changes in DNA, can we actually find better treatments for diseases? And as part of this, we kind of started this new project that we called Darwin's Dogs. It was a collaboration between my two research institutions, the UMass Med School and the Broad Institute, and an organization called IABC, which is actually an organization that's focused on animal behavior, and they work with a lot of kind of professional animal behavior people. And this was actually a really important component to the project because as a kind of academic genetics person, I'd spent a lot of time talking to people that were kind of in the academic veterinary world. But with a citizen science project where we want to go out and work with people and their dogs, we needed to work with people that actually had a lot of expertise in talking to people about their dogs. And a group of people who spends an awful lot of time talking to people about their dog's behavior are professional animal behavior consultants and things like that. And so that's been a wonderful collaboration to have. Okay, so I'm going to step back now and take you back into the mists of genomic time, which was 2005. Genomics is a very young science. So this is when we published the Dog Genome Project. This was actually the second genome project I'd worked on, and the first one I'd been really deeply involved in. I learned a ton about genomes and genome analysis. And to be totally fair, I think I probably learned most of it from Claire Wade, so I appreciate her talking this morning and kind of setting the stage for all this, because there was a lot of work that we did together kind of in the early stages of dog genetics. And so the Dog Genome Project, we basically looked at the DNA that was in one dog, and then we published the sequence of this DNA. And the wonderful thing about working in dog genetics is that you can have a lot of fun with things like journal covers. And so what we did with this journal cover is we actually have this cool little picture, and we actually photoshopped in Tasha, the dog we sequenced, into the photo of Watson and Crick in the kind of double helix structure. So it was pretty cool. And so I just wanted to quickly give an introduction. I think people have talked about this before, but what the word genome means, just because it's kind of a complicated word for a pretty simple concept. And so a genome is just all the DNA you've inherited from your parents. So you get one copy of your genome from your father, one copy of your genome from your mother, and those two copies together comprise your genome. It's basically the DNA you've inherited. And when you're kind of right after the sperm has fertilized the egg, you only have one copy of your genome because you're only one cell. But actually, by the time you're kind of an adult individual, you've got 40 trillion cells, and your DNA, your genome, has actually been copied into every single one of those cells. So you're actually carrying around many, many different copies of your genome. And the reason why we can do things like take a saliva swab to get DNA, or do a blood test to get DNA, or do a skin biopsy to get DNA, or even get DNA out of hair, is because those cells, the cells within each of these tissues, they're all carrying, nearly all of them are carrying a copy of your DNA. And so we can actually kind of do that blood test and basically pull out the sample, we'll pull out the little chromosomes, and then that's the DNA that's actually underlying it. So for the Dog Genome Project, what we did is we sequenced the genome of one dog, this dog named Tasha. The reason we did that was that the DNA of nearly all dogs is going to be almost identical. They're all dogs. They share an awful lot of DNA. And so most places in the genome, they're going to be totally the same. And so you start by sequencing the genome of one dog because it's basically like a map. It's kind of like a reference map. And then you can look at other dogs and compare them back to it. And so, we sequenced the genome of this dog named Tasha. The dog genome, it's much like the human genome. The human genome is like 3 billion letters long, so there's A's, C's, G's, and T's. The dog is about 2.4 billion, and there's about the same number of genes, about 21,000. And I just wanted to say, so how do we actually sequence the DNA? I put this together, and it actually took me a little while to put the slide together, but I actually find it quite fascinating because, as I said, genomics is a young science and it's changing really, really fast. And so you'll kind of see this new stuff coming out all the time. And so it turns out that one of the things that's changed really fast is sequencing technology. So back in ancient times, which I think is like pre-late 1990s, they did something called Maxim Gilbert sequencing, and I was at a conference recently where somebody was talking about the great old days of when they ran gels and all this stuff. The problem with it was that you couldn't actually sequence 2.4 billion bases of DNA on something that's a very slow technology, and also it was kind of radioactive. So we've moved away from that into the late 1990s when we got these fancy new automated Sanger sequencing machines, as they called them. So Sanger was the actual method of sequencing. And for this, we could actually sequence a whole genome, but actually for the human genome, it cost us about $3 billion to do the first one because we had to develop all the technology and the methods and everything. And after that, using this technology, it was probably somewhere around $50 or $100 million to sequence the genome of a single individual, whether it was a dog or a human. And that was actually how we sequenced Tasha, the first dog. And interestingly enough, we did human, and then we did mouse, and then we did dog. So dog was actually the third mammal that we actually had a complete genome sequence for. In 2007, there was this big revolution that they called next generation sequencing, which means today we have new technology and everybody's trying to figure out what to call it. And some people call it next generation. It's getting very confusing. But 2007, we had next generation sequencing, and that kind of started to change everything because all of a sudden we could do a single genome from an individual for about $5,000 or $10,000. And they've kind of been iteratively improving that technology. And I think when we sequenced my sister's dog, just coincidentally, a dog named Besco in 2014, it cost us, I think it was $1,360. And so it's kind of starting to come down into the realm of where you can actually look at the genome of an individual for something that's kind of actually, it still gets really expensive if you do want to do too many of them, but you can do it now feasibly. And so we're at this point now where we can sequence genomes. We're really good at that. The first human genome took about 13 years. Today at the Broad Institute alone, one of the places I work, they're apparently generating a new genome sequence about every 12 minutes. So that's 70, up to this point in time, I think since 2009, they've generated about 70 million gigabytes of data, and that is actually doubling every 8 months. So we no longer have a problem with actually sequencing genomes. We've gotten really good at it. And they've been doing this a lot in human genetics, and so they have this UK Biobank project where they are sequencing 500,000 individuals. And they've got another one for humans in NIH that they just announced where they're doing 1 million individuals. By doing so many individuals, you can start to find things you just can't find if you don't have very many. Dogs are lagging behind right now, but we're kind of playing catch up, but we'll get there. We just actually worked with Elaine Ostrander to post the complete genome sequences for 435 dogs on our website, so it's a start. That's just there. Anybody can go get it and start playing around with it, and we're hoping to kind of keep on building that up over time so we actually get to the scale of data that's available for human projects right now. So we're really good at sequencing genomes. That basically means determining the order of these A, Cs, Gs, and Ts. We've gotten good at it. The problem is we don't actually really know what any of it does. Which is actually really good news because, you know, career-wise, I mean, I am totally set. So the kind of, and I know there's already been some talk about the biology of genomics. I'm just going to go over it briefly. We have this central dogma in genomics that says you have a gene, the gene makes RNA, the RNA makes a protein, and the protein goes off and does things. It was a very nice story. It was really pretty. It turns out it's actually way more complicated than that. But as I said at the beginning, nearly every single cell in our body carries a copy of our genome. If it were that simple, why isn't every cell in the body exactly the same? Why do we have skin cells and heart cells and everything else that are look completely different, even though they're all carrying the same blueprint? And the other thing that's really confusing about this, as I said at the beginning, so there's billions and billions of these bases in the genome. About 1.5% of them are actually genes. We're pretty sure that another 7 to 10% of them are actually important for some reason. We don't know why. We think they're important. We have no idea why the other 90% of them are there. There are people that think it's just kind of like stuff that happened during evolution that there was no point to get rid of, so it just kind of stuck around. And there's other people that actually think it's important to have it there for spacing or because there's useful stuff that we kind of use sometimes in there. But the problem is that we actually need to figure out what the important stuff is. And so the first thing is 1.5% is genes, 7 to 10% is also important. And so that other important stuff is basically what we call regulatory sequence. So a gene does make a protein, but the regulatory sequence controls when the protein is made. So it may be turned on in a heart cell, and that's why it's a heart cell. It may be turned off in a skin cell. And so you have things around it. There are things like enhancers, where if another protein comes in and binds it, it actually turns up the expression of that gene, so you get way more of the protein being made. You got regulators and insulators will actually turn down the expression of that protein or maybe just shut it off altogether. And so those regulatory sequences are really important. They're also really interesting in the context of something like behavior, because the thing about changing the gene itself, so a change in the gene itself changes the protein. The problem with changing a protein is that you then change that protein in every single cell where it gets used. And so it might be a change that changes the color of your fur, but it might also be a change that's a gene that's incredibly important in the very early stages of embryo development. So if you change the gene itself so that you change the protein, there's a very high probability of doing severe damage to the organism. And so often you'll end up with basically they're going to be dead or they're going to be severely disabled. And a lot of the kind of really severe, the kind of syndromes that have really severe disabilities, you often see that there are changes in a gene that actually then has kind of wider effects on your whole body. The nice thing about regulatory changes is you can be much more specific. You can say, I'm going to change this enhancer here so that this gene doesn't get turned up as much. But it turns out this enhancer is actually only used in skin cells. So by changing it, I'm only changing the function of this gene in skin cells. And that's how you actually end up controlling not just whether something is a skin cell or a heart cell, but also particular phenotypes about it. So what I've actually been focusing on is the first problem. So 1.5% of the genome is actually genes, another 7 to 10% is important. We've got a set of animals that are different in some way. How do we figure out what pieces are actually controlling that thing that we're interested in? And it's actually not a particularly complicated way that we do this. We basically take a whole lot of dogs that have a trait that we're interested in. In this case, it's white coat color. We take a whole lot of dogs that don't have the trait that we're interested in. So in this case, they're solid dogs, and we compare them. And any two dogs will have differences between them, but if you see a consistent difference between the solid dogs and the white dogs in their DNA, and you say all these dogs, the dogs all look different at this point, that's a suggestion that it actually has something to do with the trait you're interested in. And so as I said, every dog will have differences between it. I just put this in here because people ask me about this sometimes, is how different are two dogs. Two dogs from two different breeds have depending on what kind of differences you're looking at, but it's about one change every 1,000 bases. So 99.9% of their DNA will be exactly the same, and they'll have a change in about one every 1,000 bases. Dogs within a breed are going to be more similar to each other because they're more related to each other. But the differences between two dog breeds, this is actually almost exactly the same as what you would expect to see if you looked at compared two unrelated people. So it's not that the dogs between two different breeds are really, really different from each other. It's that dogs within a breed are very similar to one another. And so I'm just going to, I use this term SNP, so I put this in there. There's this fancy word called a SNP. It's not actually particularly complicated. When you look at DNA, you can sequence all the DNA, which, as I said, is still expensive and used to be really expensive. Or you can use strategies that say, I'm going to only look at points in the DNA that I think are going to be different between dogs. So I've observed differences between dogs at this position, so it's a good place to look for differences. And so we call those positions single nucleotide polymorphisms, which is a very fancy way of saying a one-letter change. And so, for example, if dog 1 had a G at a position and dog 2 had an A, that would be a SNP, or a single nucleotide polymorphism. Most differences, as I said, they're going to fall in that 90% of the genome where we don't think the sequence is important. Just because a dog has a difference in a particular point doesn't mean it's actually doing anything. It's just a difference. It's just a marker for some, for what it's, probably inherited from one of its parents. So it can be useful for tracing ancestry, but it's not changing the dog in any way. So what we'll do is we'll gather a whole lot of dogs, either with or without the trait we're interested in. We'll do a swab with saliva. We used to do blood samples. We'll extract the DNA out of it. We'll run it on a fancy machine and basically test thousands of these SNPs. And actually, we used to do thousands, and with the kind of most recent technology, we're looking at millions per individual. And for each SNP, we're just going to go across. We got our solid dogs and we got our white dogs, and we're going to say, at this position in the genome, how different are these two groups of dogs? And so you just go across the whole genome and you ask that question over and over and over again. And what you're looking for, as I said, is your SNP correlated with the trait. So I kind of put together this little example here. So if you look at this position, it turns out there's a little bit of a difference. There are more Gs in the solid dogs and there are more Ts in the white dogs, but there's still, it's not a perfect match to the two traits that the two different phenotypes that we're looking at. And the same thing here, there's more Ts in the solid dogs and more Cs in the white dogs. It's not perfect. And then at this position here, we see 100% correlation. So the solid dogs look completely different from the white dogs. And that's kind of what we're looking for. In that position, we get a really high score in the test that we use. So we actually did this in coat color. This was the first trait that we looked at, and we looked at it partly because Tasha, that boxer that we sequenced the genome of, was actually heterozygous for this trait, meaning that she had one of her parents, she'd inherited the solid version, and from one of her parents, she'd inherited the white version, and that meant that we knew somewhere in her genome we'd actually captured the mutation we were interested in. So the way that the inheritance of coat color works in boxers is that if you get two normal copies of whatever the gene is that's controlling it, then you're going to be solid. If you get a normal copy and a white version of that gene, then you're flash, so you're white on the front and you're solid on your back. And if you get two copies of the white version, you're almost completely white. You'll sometimes have a patch of color, often on your head, and about 2% of these dogs are also deaf. And so we took 9 solid dogs and 10 white dogs, and as I said, we had Tasha, the dog we sequenced, who was flash. And we did the test I'm talking about. We looked at SNPs all the way across the genome. In this case, I think it was about 20,000 of them. At every single SNP, we just asked how different are the solid dogs and the white dogs. And so the graph I'm showing you here along the x-axis along the bottom is just the position in the dog genome. The dog genome has 39 chromosomes. So that's why there's all those numbers along the bottom. And then on the y-axis, I'm just showing you how different are the solid dogs and the white dogs. And you can see we went across the whole genome and asked for every SNP, which is just one of those dots, how different are they? And then we hit chromosome 20 and we got a really, really big peak. And it turned out that was statistically significant. And we went and looked at it, and it was over a gene called MIDF. And MIDF was actually it was an interesting gene because it was known to impact coat color, but they looked at it in dogs before and hadn't found any differences in the gene itself between the white dogs and the solid dogs. And so they couldn't really figure out what was going on. But it turns out that this is one of these genes where it's a problem to actually change the gene itself because it does many different things. And two of the things that it's important for is that if you start making the gene all the way at the beginning, which is called the A version, then you get a version of the protein that you have to have healthy eyes. If you start making the gene a little bit further in, so start making the protein a little bit further in, at what they call the M start site, then you make a version of the protein that you have to have in order to have normal pigmentation. The boxers have totally fine eyes, so it's very clear we haven't damaged this gene in any way that's going to disrupt the protein so that you can't make healthy eyes. We've only changed it in a way that's going to affect the coat color. And it turns out that when we looked at bull terriers, which have the same phenotype, and narrowed down exactly what the position in the genome was that we thought was responsible. And it turns out that it wasn't in the part of the gene that makes the protein, which is the part that I've colored dark blue here, but it was right next to the M start site. And that was kind of a red flag that what we're looking for is a regulatory change. So we've somehow disrupted how much this protein gets turned on, how much this gene gets turned on or turned off. And actually, it turns out that this makes perfect sense. So we found a couple of mutations upstream of that gene that we think were probably responsible. One of them was actually a change in the length of the DNA. So the DNA was like 4 letters longer in the white version of the gene. And it turns out that doesn't sound like a lot because 2.4 billion letters of DNA, but it turns out that DNA, actually each turn of the DNA is 8 bases long. So if you insert 4 letters, you actually change the positioning of things in the DNA relative to one another, which actually could affect that thing where another protein comes in to bind something to actually turn on the gene. And so it may be that we disrupted the binding of something else to the DNA at that position. And the other thing I really like, and the reason I keep talking about this story, so it turns out that what MIDF is doing for pigmentation is that it actually controls the migration of pigment cells during development. So when you're developing as an embryo, your pigment cells start on your neural crest, which turns into your spine, and then they migrate around the whole rest of your body. So the solid dogs' migration proceeds completely normally, so they're completely pigmented. If they have a patch of white, it's usually right on their chest, because that's the last place it gets filled in. The heterozygous dogs, the ones that have one normal copy and one white version, the pigment cells can only make it halfway around. And so they have solid on their back, but they've got white fronts. And then it turns out that you have to have pigment cells in your ears in order to be able to hear. So in the white dogs, most of the time they must have just enough pigment cell migration that the pigment cells get to their ears. And if you see pigmentation in the dogs, in the white dogs, you normally see it as a big patch on their head. So that's one pigment cell that has managed to migrate and then it kind of replicates out again. But in 2% of the dogs, apparently the melanocytes, the pigment cells don't even get as far as the ears, and that's why those dogs are deaf. And so the other reason I really like this story is it kind of illustrates why we're studying dogs. And so, as I said, they'd actually studied this protein MIDF for years. And they'd broken it in mice, because that's what they do in lab mice. They go in and they'll delete the protein, or they'll make it so the gene totally doesn't work or something. And that did end up with white mice. But they also got mice that were deaf and blind and mostly dead. And so that wouldn't have worked in dogs because dogs are pets and we live with them, and we wouldn't have wanted dogs that were white but also deaf and blind and also dead. And so by studying what we call a natural model like dogs, something that's actually in the world with us, we can find these kind of subtle mutations that are affecting the regulation rather than these kind of big things which we would kind of artificially put into mice. And especially in terms of studying diseases like cancer that actually involve these kind of regulatory changes, dogs are going to be a lot more similar to humans than something that we've kind of created in the research lab. And so it kind of illustrates why you would want to study something like a natural model, even though it's going to be much more complicated than dealing with something you've kind of made yourself. So we did a bunch of these different kind of single gene traits, and this is something that many, many other people in the dog world have looked at as well. And it turns out we got pretty good at that, and the methods are relatively well established for finding the genes that are responsible when you have something that's caused by a change in a single gene. So we decided, of course, being scientists, that now we would move on to something way harder. So we moved on to what they call canine compulsive disorder. And so the interesting thing about canine compulsive disorder, and I didn't know this when I got into it, was that it turns out that it's remarkably similar to human obsessive compulsive disorder. And so compulsive disorder, OCD and CCD, are defined as doing an otherwise normal behavior way too much. So dogs don't wash their hands because that wouldn't be normal for a dog, but they do something that a dog could do normally, but they do it way too much. And they do it to the point in time where it's actually going to impair their quality of life. And kind of a description I've heard of it is it's the kind of thing where if you visited somebody that had a dog that had a compulsive disorder, you might not know there's anything wrong, but if you live with that dog and you know how much they're doing it, you're aware of how disruptive it is to their life. There's a similar age of onset, so humans that have the more severe form of OCD often kind of get it in early adolescence, and it's the same in dogs, somewhere between 6 and 12 months of age. And it's highly heritable in both species. So in dogs, we see that certain breeds tend to be prone to it, which is an indication that genetics is involved. And in humans, if one of your parents has OCD, you're much more likely to get it. I think it's what they call about 40 or 60% of it's kind of heritable or caused by genetic factors. And this actually points to the fact that it's not just that it looks the same in the clinic, but that actually the same things might be going on in the brain. So it turns out we treat dogs and humans with the same drugs. So we treat them with SSRIs like Prozac, and they work just as badly in both species. So about 50% of people and 50% of dogs get some kind of relief from these dogs, from these drugs. And the reason I bring that up is that's the other reason we really need to understand what's causing these diseases. And so especially with psychiatric diseases, there's drugs available. Many people don't actually have good options. They don't respond to the drugs, or the drugs have such terrible side effects that they can't take them. And the reason why you would actually want to understand what's causing these diseases on a genetic level is that might actually tell you more about what's going on in the brain, and you might be able to design a therapy that, rather than affects the signaling of serotonin across your entire brain, actually targets whatever pathway is actually responsible for the compulsive behavior. And that may have fewer side effects and be more effective. So if I can actually move to the next slide. So we did exactly the same thing we did with coat color. Took a bunch of Dobermans that had compulsive disorder and a bunch of Dobermans who didn't, compared their genomes, looked for differences. You can tell that this is a complex trait, one that's controlled by many genes, because it's not as clear of a signal as what we saw with the coat color. And we also needed a lot more dogs even to get this kind of signal out of it. But we did get a really nice, what we call a statistically significant signal on one chromosome, chromosome 7. We got very lucky because there was a gene in the peak. Usually there's no gene, and then you have to say, really, it's interesting, even though I have no idea why we have a peak there. But in this case, luckily, there was a gene in the peak, and it was a really, it was a gene that seemed to be kind of obviously relevant to the disease. So CDH2 is it's a neural cadherin, so it's basically a protein that's involved in linking up the presynaptic and postsynaptic membranes of certain synapses in the brain. And it actually seems to be found a lot in glutamatergic synapses, and it's involved in learning and memory. The other thing we found in this study that really kind of guided a lot of the stuff I've been doing since then was that we identified one of the major problems with studying a disease within a breed. And so we looked in the Dobermans, and that was where we found the CDH2, which was different between the dogs that had it and the dogs who didn't. But we also looked at regions of the DNA where all the Dobermans looked exactly the same as one another. Because they're closely related, there's less diversity, so you have these chunks of the genome where every dog in that breed looks exactly the same as every other dog. And the second largest region that we found like that in the genome on chromosome 17 actually had this other gene in it called CTNNA2. And CTNNA2 actually encodes a protein that's involved in linking CDH2 to the presynaptic and the postsynaptic membranes. We can't actually detect an association of CTNNA2 to a risk of compulsive disorder in the Dobermans because there's no difference between the cases and controls. But it may be that that's one of the reasons why a breed could be at a particularly high risk of a disease, because basically all of the dogs have a risk factor. It's a risk factor, so that doesn't necessarily mean you're going to get the disease, but it means you're at a higher risk of getting it. And we actually went on and studied 3 more breeds and compared dogs that had compulsive disorder to dogs who didn't, and saw that in those other 3 breeds, CTNNA2 did seem to differ between the compulsive disorder affected dogs and the healthy dogs. So this is kind of where we got into. And you think about this and everybody's like, oh, you figured out this whole compulsive disorder thing, and it's like, no. We only studied 1 breed, and then we added on those extra 3, but we only looked at the whole genome of 1 breed, those Dobermans. And that CDH2 locus that we found, even within the Dobermans, is probably only explaining somewhere between 5 and 8% of the risk of getting this disease. So this is not even close to the whole story. It's just a very kind of the first initial information about it. And I put this in here because this is, the coat color. It's like if you have this, then you're solid, and if you have this, if you're white. That's not the way behavior works. And so what we actually saw was that the unaffected dogs were much more likely to have 2 of what we would call the normal. We call it the kind of wild type. It just means the version that we think most dogs have. 2 copies of the kind of normal version of the gene. And the ones that were flank suckers, that's kind of an intermediate phenotype, so they had a compulsive disorder, but they only had the one behavior they were showing, were less likely to be purple-purple and more likely to carry that red allele. And then the ones that had multiple compulsive behaviors that they were showing, which we think are the most severe cases, actually seem to be more likely to carry that red one and less likely to carry the purple one. And another way of showing it is just that if you were purple-purple, 10% of the dogs that had 2 normal versions of the gene had compulsive disorder, 25% of the dogs that had 1 normal version and 1 of the altered versions had it, and then 60% that had 2 copies of the altered gene had it. So it's a risk factor. It increases your risk, but it's not you can't actually define which dogs are going to get it based on their genetic at this locus alone. And I put this in actually fairly recently because it's something that I've been thinking about a lot now that genetic testing is a lot more available for dogs. And so for this study, and for the vast majority of dog genetic studies out there, they've done what we just did. We found a group of dogs, we had affected and unaffected, and they found genetic variants that were correlated with the trait they were interested in. It's a start, but it really is only the first step, and you cannot go from this to a genetic test for a disease. You've completely missed the next step, and that's that you need to test whether this variant is useful for predicting disease. So you have to go out and find an entirely new set of dogs, one that was not involved in your initial study. You need to predict using genetics whether this dog is going to have a compulsive disorder, and then you need to go in and see which dogs actually have a compulsive disorder and see how well your prediction actually works in defining which dogs have the disease. And then, because we're working in dogs, and if we did the study in an initial breed like the Dobermans, you then have to repeat that for all the other breeds and for mixed breed dogs, because we don't know how the genome background within a breed is actually affecting the way that marker works. So it's actually a very long process, and I think it's important to kind of keep in mind that a lot of times they've only kind of done that first piece, and just because you found a correlation, that doesn't mean you actually have a test that can predict a risk of disease. And then I also put in this slide just because this kind of summarized it all, because people kept asking me about it. So the things you really have to be careful of when you're thinking about genetic testing in dogs is, first of all, it's completely unregulated. So we don't know when you get a result back for a genetic test how good that result is necessarily. And it would be, probably different organizations have different ways of validating the test, but it's important to know how they're validating it. There was a paper that recently came out in the human kind of health world where they looked at genetic tests that were being done looking for mutations in cancer genes in humans, and they found out that in 40% of the tests, the result that the people got back was actually just wrong. They used another method of looking at the DNA and found out that they just had the wrong answer. So that was kind of a red flag for you. You have to make sure that they're actually validating how they're actually detecting the mutation to begin with. They're often developed in small numbers of purebred dogs. They often haven't tested how well the variant predicts disease, just that it's correlated with it. And finally, and a lot of times they're like, take the genetic results to your vet and see, ask them about it. Veterinarians are not trained in clinical genetics. It's an incredibly complicated kind of field of research that's changing all the time, and they often don't have the information that you're going to need to actually interpret those tests. So that's then like, why is this useful? A lot of times you think, well, let's do a genetic study and find markers so we can predict which diseases are, which dogs are going to get sick. And that comes back to that first part, where the actual idea here is to understand the underlying biology, to understand how these diseases actually work. And what we have found out, as I said, we found this CDH2 in the Dobermans, and it turns out that they've been doing genetic studies in humans too, and they found a whole set of other genes that were involved in the same kind of glutamatergic synapse pathway as what we found in the dogs. And so we kind of took a step back and looked at genes that had been found in dogs, genes that had been found in humans, and genes that had been found in mice. And in this little illustration here, we got the two synapses, the presynaptic and the postsynaptic synapse. And then we just marked with little icons, the human genes with a human head, the dog genes with a dog head, and the mouse genes with a little Mickey Mouse head. And what you can see is that by studying many different species, what we're actually doing is getting a much more complete picture of the pathway that might be involved in this disease. So if we only had 2 genes, those genes might be doing a lot of other things in the brain. But if we can say that this whole set of genes is involved, that might actually be pointing us more specifically at a particular pathway in the brain. And so if we could actually define these pathways better, we may be able to develop new targeted therapies for actually treating these diseases. And so that's kind of where I got to in the first half. And in the second half, I'm going to kind of talk about this is what we've done so far and how are we actually going to make this better going forward by getting much bigger sample sets and working with people and their pets to understand much more about what's going on, at least as far as owners perceive it in the minds of their dogs.